As you know, Delbecq and Claude S. Ruppert, they could not explain the chemical basis of the phenomena of photoreactivation. The problem of the chemical nature of photoreactivation was resolved over next few years when investigators came to know that UV irradiation induces the formation of cyclobutane pyrimidine dimer in DNA. Before that, the scientists, they were not aware of this dimer formation. They didn't know what happens after UV irradiation in the DNA. But when they came to know that cyclobutane pyrimidine dimer is formed in the DNA, and then they came to know the chemical basis of that reverse, that photoreactivation. Further studies showed that the photoreactivation enzymes reverses the UV induced damage by using the energy provided by blue light, and this blue light ranges in wavelength between 350 to 450 nanometer, and this energy is used to disrupt the cyclobutane ring. You know, a ring is formed between two adjacent bases after UV irradiation, and during the reversal of this damage, this ring is disrupted. When it was recognized that the photoreactivation enzyme catalyzes the disruption of carbon-carbon bonds, it was given a more descriptive name as cyclobutane pyrimidine dimer photolyase. So here you can see in this picture, this is normal DNA, and when this is exposed to UV light, a dimer is formed. Here you can see this is a dimer. So a dimer is formed, and when in the presence of photolyase enzyme, this is CPD photolyase, this ring, these bonds are broken and normal DNA is restored. So this is the action of photolyase. The bacteria that lack CPD photolyase cannot repair cyclobutane pyrimidine ligands through photoreaction, thus cannot reverse the UV induced damage in DNA, because these are CPD photolyase which reverses the damage, and if CPD lyase is not present, how an organism can reverse this UV induced damage? CPD photolyases are present in a wide variety of organisms, including bacteria, archaea, plants and animals, but unfortunately these enzymes are not present in humans and other placental mammals. That's why if we are exposed to UV light and if our DNA is damaged, we cannot repair that damage. That's why the people which are exposed more to UV light are more prone to cancers. There are these CPD photolyases are monomeric proteins, and they range in size from about 450 to 550 amino acid residues. These enzymes contain 450 to 550 amino acids. All CPD photolyases have two domains, and these two domains are designated as N-terminal domain and C-terminal domain. A light-absorbing pigment or chromophore pigment binds to each domain through non-covalent interactions. So there are two domains, C-terminal and N-terminal domain, and a pigment, chromophore pigment, binds to each domain. So there are two chromophore pigments, and they bind to these domains through non-covalent interactions. This chromophore factor acts as a photoantenna to capture light with wavelengths that would not otherwise be available. Although the light is available, but the cells cannot absorb that light, and if the light is not absorbed, photoreactivation cannot take place. So these chromophores, they act as photo antenna to receive the light, to absorb the light, and to transfer that energy to the enzymes.